patterns and correlated patterns from the data. So another strategy is to embed biological and clinical insights about the phenomena of interest directly into the model. So this information can be derived from just the structure, the topological nature of the data that we're studying, insights from our collaborators, or even the broader scientific literature. And then finally, we've developed new training strategies that are geared towards learning from weak labels and missing data. So application-wise, my lab works across what I call the scientific spectrum, from addressing foundational neuroscientific questions that, with models that can potentially be applied to many, many different types of studies or many different studies, to translational applications of biomedical signal and imaging data, to exploratory directions for understanding the brain and brain-body interaction and cognition. So for the rest of this talk, I'm going to focus on two ongoing projects that tackle these domain challenges in different ways, and also are kind of different sort of in terms of where they lie on this application spectrum. So I'll start with our work on imaging genetics. So as a preview, our work here is actually going to be, our approach is going to be to design our deep network architecture using a priori hypotheses from the literature. So the starting point of imaging genetics as a field is to note that neuropsychiatric disorders, so like autism and schizophrenia, they have two complementary viewpoints. So on one hand, they're associated with behavioral and cognitive deficits, such as impaired social responsiveness or memory, working memory deficits. And in some sense, these deficits manifest through altered patterns of neural activity that we can capture through a neuroimaging modality. On the other hand, these disorders tend to run in families, meaning there's a heritable component. So it's very likely that there's a genetic underpinning that's influencing downstream biological pathways and then leading, or at least helping to lead, to the observed phenotype. So imaging genetics is an emerging field that really tries to link these two perspectives to identify more robust and targeted biomarkers. And so before diving into the model, I'm just going to quickly go over the data that we're working with, and I'm sure this is familiar to many of you. So on the neural side, we're looking at fMRI. So functional MRI is used to measure the brain dynamics. So it's essentially a 4D acquisition with a relatively high spatial resolution, so between 2 and 5 millimeters cubed, but a fairly slow sampling rate, so between 2 and 5 seconds. So as you can see on the slide, we can think of fMRI as a collection of volumes over time. And alternatively, every spatial location has an associated time course. So many fMRI studies, or at least traditional fMRI studies, relied on carefully designed paradigms to activate specific neural processes. So for example, if we're interested in visual response, the subject might see a sequence of images or pictures while lying in the scanner. Now, once acquired, the fMRI is analyzed using a multivariate regression framework known as a GLM. So here, effectively, we're going to model the time series for each voxel as the product of a design matrix that encodes the stimulus timing information and other types of noise and confounds as well. And then the second element of that product is a vector of activation coefficients, so those betas, which we can estimate from the data. And then we can use these activation coefficients to construct a contrast map that really sort of quantifies the relative change in brain activity associated with the task. And this contrast can be aggregated across different brain regions to form an imaging feature vector. So from the model's point of view, that imaging feature vector is going to be one of the inputs. Now, in contrast, genetic variation is often quantified using single nucleotide polymorphisms, or SNPs. So SNPs sort of fundamentally are substitutions at a particular loci on the genome. So for example, if most people have a CG pair at a particular loci, then a SNP would be an AT pair there instead. And SNPs are encoded categorically. So 0 indicates no change. So both alleles have the conventional pattern. 1 denotes a change on a single chromosome, and 2 denotes a change on both chromosomes. Now, the most common analysis methods for SNPs is called a GWAS, or a genome-wide association study. So at a high level, so we're going to use a sample, like some independent sample of tens of thousands of subjects, and compare the relative frequency of each SNP between a set of patients and a set of controls. And this gives us a p-value for the genetic effect or the genetic association between that SNP and whatever the disease is that we're studying. So in most studies, we might use a stringent p-value threshold and extract like sort of a reduced subsets of statistically significant SNPs. And similar to fMRI, these subselected SNPs can be organized into a genetic feature vector, where each element is like the effect size of that particular SNP. Right? So again, from the modeling standpoint, that feature vector will be one of the inputs. Now, in terms of prior work, there are a few broad categories of imaging genetics methods. So the first is a linear regression framework that tries to map that genetic feature vector onto a single imaging feature. So maybe change in activation in a particular brain region. Now, while this has been one of the most popular approaches in the literature, it provides a very limited perspective of that neural functioning, because again, we're picking a single feature, so maybe cortical thickness or some activation change in a single location. And in addition, the phenotype, so case control information, is not really factored into that linear regression model. It's done post hoc. So like we're effectively going to sort of compare whatever the coefficients we get in cases versus control. And so the identified relationships might not actually be informative of the disease status. Now, the second method is to effectively learn a projection that aligns the imaging and genetic feature vectors into some kind of shared low-dimensional space. And the common methods here include canonical correlation analysis or parallel ICA. So while this approach certainly provides more information than a single regression, because we're using like all of the imaging and all of the genetic features, it really relies on kind of a pair of data sets. So it can't capture multiple snapshots of the brain. And also, again, the disease information is post hoc. So it doesn't actually go into this correlational type of analysis. Now, there are two methods, broad methods in the literature that focus on combining imaging, genetics, and diagnosis into a unified framework. The first is a Bayesian model proposed by Botmanglic et al., which uses kind of a set of parallel logistic regressions. So one regression to map the SNPs onto the sort of set of neuroimaging features, and another to map the neuroimaging features onto the diagnosis. Now, while this is certainly a seminal contribution, the approach doesn't capture direct genotype-phenotype relationships, and it instead assumes everything goes through whatever that particular brain modality is. And once again, it does rely on kind of a single snapshot of the brain. The second approach is actually an earlier work, which I'm not going to talk about in this talk, which uses a couple dictionary learning framework or a couple of dictionary learning models to project the imaging and genetics data onto a shared low-dimensional space, analogous to a CCA. And that space is also predictive of the phenotype. And so everything is optimized jointly. So while we've demonstrated improved phenotypic prediction with this model, it's also designed for a single imaging modality and a single genetics modality. And then finally, something I've swept under the rug for the last couple of slides, all of these methods really require this really low dimensional genetics extraction. So you have a very stringent significance threshold, so you're getting on the order of 100 SNPs or so, right? And so that's actually throwing away a lot of information about the genome and how they might relate together. So in our current work, we're actually going to use deep learning to overcome some of these limitations. So the overall framework is very similar in spirit to the coupled dictionary learning that we had explored earlier, but all of the operations will be effectively non-parametric and performed by neural networks. So this work was done by my current graduate student, Cheyenne Gosal, who's at Johns Hopkins, and our collaborator, Anand Mate, who is at the Lieber Institute. So the secret sauce of our framework is how we encode the data. So what is the actual sort of function or network we're going to use to extract this information? So we'll start with the imaging, because that's the most straightforward. So as I described earlier, we have an fMRI feature vector where each element corresponds to, let's say, average activation or some particular metric in that brain region. Now, our hypothesis is that only a subset of brain regions are actually informative for the downstream data fusion. It's not necessarily the entire brain. And this is very similar to a sparsity assumption or a sparsity prior. And we're actually going to automatically learn kind of the importance of each of these regions using dropout. So here, this vector Z is a stochastic dropout layer where the irrelevant regions are more likely to be masked and the important regions are more likely to be preserved and flow through the network. Now, in order to learn these importance probabilities, we're actually going to model Z as a continuous relaxation of a Bernoulli random variable, so in the equation, the key things are there's a probability vector, these P's, which effectively are the feature selection probabilities, so they act like a feature importance. And then the random variable U provides stochasticity so that as we're training these models, we don't keep falling into and getting trapped in local minima. So all of this we can optimize using a standard backpropagation. And kind of the important thing that we want to extract is that P vector. So that tells us how informative each region is. Now, following the learnable dropout layer, we use a simple cascade of fully connected layers to obtain the low-dimensional encoding. So if we go back to our model overview, we can actually use this same encoding for any number of imaging modalities in our study. So if we have multiple fMRI paradigms, we just add multiple branches. If maybe we want to look at cortical thickness or some sort of structural measure, we again add branches for that data as well. So we can sort of combine an arbitrary amount of information here. And then the decoders are simple up projections. So it's almost like a mirror image of the encoding. And for the most part, that decoder acts as a regularizers, just to make sure that the information is being preserved in the network effectively. So that takes us to the question of how do we encode the genetics data? And here's where we're going to draw inspiration from the biology literature. So stepping back, we can recognize that the SNPs, so these like isolated points or loci on the genome, are not really working in isolation. Instead, they either lie within or they're proximal to different genes, which are then encoded and transcribed. And even these genes also don't work in isolation. We don't just produce a protein and it floats around randomly. Instead, they contribute to more and more complex biological processes. Right? So our strategy is actually to design the encoder to mimic a hierarchical ontology of biological processes. And so this is actually kind of an established ontology within the cellular and computational biology literature that was validated using laboratory experiments to try and confirm these relationships. So an example pathway through the ontology is shown in the slide, starting at more specialized processes like neurogenesis and then going to more sort of general processes like cellular development, et cetera. And just in terms of the modeling itself, if we look at that ontology, there's two salient features. First, there are the processes themselves, and those are going to represent nodes in our deep network. And then there's the relationship or the links between certain processes, and we're going to represent them by edges in the network. So in terms of the encoding, our first step is to group the SNPs according to the nearest gene and aggregate the information into a single gene score. So this is a standard procedure in computational biology, and it's actually based on a weighted summation of genetic risk. It's a non-deep learning operation, so I won't spend too much time talking about it, but we can consolidate that SNP information to about 14,000 genes. Now we're going to learn a projection of these gene scores onto the biological processes specified by our ontology. So these processes are going to correspond to hidden nodes in our graph network, in our deep network. And the operation is analogous to a sparse perceptron layer where only a subset of the input nodes are connected to each hidden node. We then use graph convolutions to model the information flow between the biological processes as governed by the ontology. And as an added layer of interpretability, we're actually going to learn an edge attention weight. So we're effectively going to learn a weight on each edge of the network that sort of quantifies and captures how much information is flowing there. And these edge attention weights are subject specific, so then we can go back and try and understand how is the information flow different between patients and controls. And finally, we use hierarchical pooling to coarsen the graph at each convolutional stage until we reach a final encoding at the root node. So going back to the model overview, our genetic decoder actually uses kind of the flipped mirror image, sort of mirror image network, and it's a set of graph deconvolutions based on the same ontology. And then finally, our latent fusion is actually very embarrassingly simple. So basically, we're going to just take it as the average of all of the encodings, where here MN corresponds to the number of modalities available for that particular subject during training. So if we only have 2 modalities available, let's say 1 fMRI and the genetics, MN would be 2. But if that particular subject was able to get more paradigms, then MN might be 3 or higher. And so that allows us to effectively handle missing neuroimaging data, because we can just freeze the branches of the network that don't have the associated modality, but still update the rest of the network and sort of maximize the information. And then finally, our classifier is a simple cascade of fully connected layers. And as an aside, we can easily adopt this same framework for a regression task just by kind of changing that classification module to a regression module instead. And then our loss function during training consists of 3 terms. So first we have a reconstruction error for each branch. So that's how well the autoencoder can actually go from input data and reconstruct that data. Then we have the prediction accuracy, which in our case, because we're doing classification, is binary cross entropy. And then we have a KL divergence term that encourages sparsity of the selected imaging feature selection probabilities. So we validated our framework on a population study of schizophrenia, and this study has 2 fMRI paradigms. There's an NBAC paradigm that assesses working memory, and an SDMT paradigm that queries visual encoding of neutral and aversive stimuli. We use the BrainTome Atlas to define 246 cortical and subcortical regions, and our fMRI feature vector is the average contrast within each of these defined regions for each paradigm. For genotyping, it was done by our collaborators using an Illumina chipset with Plink for pre-processing. This gave us about 100,000 SNPs, which we grouped to the nearest gene with a 50 kilobase pair overlap. The group SNPs were used to create about a 14,000 dimensional gene score, so that was the input to our model. And then the projection weights were obtained from a held out data set, so there was no bleeding of training and testing information. Finally, our data set consists of 1,848 subjects with just the genetic data, and we use that for pre-training, that sort of large genetics ontology model. And then we have an additional 210 subjects with both imaging and the SNP data. But as you can see on the table, not every subject had each imaging paradigm. So there's actually quite a bit of missingness in terms of how much data we have for NBAC, SDMT, and genetics. So the first thing we did was quantify the predictive performance via a repeated tenfold cross-validation. And so this table is just representing the patient versus control prediction accuracy on the testing data set. So to parse this a little bit, the first row is a classical approach. So here we are going to use parallel ICA to construct a low dimensional representation and random forest for classification. And we use a data imputation strategy to handle missing data, which might actually contribute to that poor performance. The next 2 rows use our same autoencoder classifier network, but we are replacing the genetic ontology with a feed-forward network with the same number of tunable parameters. So we're just breaking the graph, but saying we can allow the same amount of parameters to flow through. And as you can see, regardless of whether we pre-train the genetics branch or randomly initialize it, our performance is capped around 0.67, 0.68 AUC. Similarly, we can decouple the imaging and genetics branches, so just predict based on imaging and just predict based on genetics. And once again, the predictive performance is a bit capped. And then finally, we get a lot of synergy when we actually combine all of this together. So our final model, which we call Guide, achieves the best overall performance, and it kind of underscores the synergy between imaging and genetics, assuming you're mining the information correctly. And it also indicates that this graph-based information propagation, or sort of the graph-based learning, carries a lot of value because it allows us to more accurately sort of extract this information on small data sets. Now, beyond predictive performance, one of our goals was interpretability. So on the imaging side, we can visualize that feature selection probability vector, so that vector P, that from the learnable dropout layers. And so for NBAC, we can see areas in the frontal cortex, like the MFG and the IFG. In SDMT, we note that the cuneus and areas of the occipital lobe tend to play more of a role. And that makes sense because they're more used in visual processing. We can also do a semantic decoding of these regions using the Neurosynth repository. So Neurosynth is a publicly available resource, and it aggregates sort of keywords and activation coefficients across pretty much the entire fMRI literature that you can extract it from. And it's almost like a meta-analysis of the literature. So what cognitive processes, or at least descriptive terms, are used to define or describe certain activations across the literature. So the meta-analysis reveals a great deal of overlap between the two paradigms in terms of the regions that are important and the concepts that are associated, but we do see some key differences. So there's vision and action for SDMT, and there's also memory retrieval that's sort of more unique to NBAC. Now, for genetics, we can actually evaluate the information flow across these biological processes, which is actually a new way of understanding genetic data in and of itself. So the analysis we do here is we first trace all pathways from leaf to root node through these networks, and then we compare the distribution of the learned attention weights along these pathways between patients and controls. So in total, we extracted 152 pathways that are statistically different between patients and controls and appear in at least 70% of the repeated cross-validation trials. We then perform a semantic embedding of the terms that describe the biological processes associated with, so these descriptive terms associated with the nodes in our hidden network. And then the figure illustrates kind of a data-driven clustering of these embedding vectors. So we notice that a few of the clusters are such as calcium ion transport, action potential, and sensory perception have been previously studied in schizophrenia and seem sort of reasonable, but our network also identifies new processes and pathways which can be used to guide future exploration. So either sort of more fine depth analyses of the associated SNPs or even laboratory experiments. So finally, I want to just note that this imaging genetics framework can be applied to a broad range of neuropsychiatric disorder where you have some neuroimaging and some SNP information. In fact, we're starting to do this with autism just to see, again, what are some of the differences that we see here as well. And then finally, outside the neurospace, effectively what we have is this sort of really cool tool to parse genetics information. And then we have, in our case, we're using sort of fMRI paradigm. So we have a particular feature vector, but we can think about changing that imaging encoder and actually applying this model outside of the neuro realm. So let's say if we have like images from sort of pathology images or from cancer diagnosis, we can try and construct a sort of a different type of imaging encoder and imaging decoder and still do that same fusion. So sort of with that project sort of wrapped up, I'm going to turn to a completely different direction. So this is a more translational endeavor for epilepsy. So and as a preview, the approach here is actually going to construct a deep generative hybrid to blend classical and AI techniques. So to set the stage, epilepsy is one of the most common neurological disorders worldwide. It's characterized by recurrent and unprovoked seizures, which have a devastating impact on patient health and quality of life, and even like a very great burden on caregivers as well. Now, while epilepsy is usually treated with medication, about 20 to 40% of patients are considered medically refractory. So that means that their seizures are persisting despite different combinations of drugs that they've tried and been given. So alternative therapies for these patients rely on clinicians being able to identify where and when our seizure is starting in the brain. Now, kind of sort of medical refractory epilepsy care is a very, very long pathway, but EEG is arguably the most important diagnostic tool for epilepsy. So it's the first and foremost modality pretty much connected on all of these patients. So here, a patient is admitted to the hospital, and then the EEG electrodes or sensors are adhered to their scalp, and then the patient is monitored for several days as they're continuing to have seizures. And then the data sort of collected across these days are evaluated after they're discharged. Now, what's really interesting in epilepsy is that despite decades of research into signal processing and machine learning tools and methods for EEG, clinically, in the clinic, sort of this EEG data is analyzed almost entirely by visual inspection. So what I mean is that an epileptologist will effectively stare at recordings like this one that are collected over many, many hours. And here on this slide, the channels are stacked vertically. The bottom one is the heartbeat, so we can ignore that. Channels are stacked vertically, and then time proceeds on the x-axis. And so based on effectively staring at these images, the epileptologist will try and identify where and when the seizure is starting. Now, I like this slide a lot because it actually highlights why the problem is so difficult and why many automated methods fail. So if I were to tell you that a seizure is starting somewhere on the slide, most lay people, including myself, and most AI algorithms, I would argue, would focus on the most salient features of the data. Now, the salient or really prominent characteristics are arguably this kind of really high ripple, high intensity signal somewhere in the middle. And then there are these weird oscillations kind of on the right that you see that are distinctly different. And it turns out both the AI and us would be wrong because all of that is artifacts. So the middle is just muscle artifacts, and then eye movement can cause sort of significant distortion. And in fact, the seizure signature itself can be very subtle. So for this particular recording, the seizure was annotated as this rhythmic oscillation in that first channel. And more than this, the seizure signature itself can vary across patients tremendously, and even across seizures of the same patient. So it is a very complex problem. With that said, there's been plenty of methods developed for seizure detection from scalp EEG, and most of them follow a standard machine learning pipeline where raw EEG comes in, is pre-processed and chunked into small time windows, and then features are extracted from each time window, and each window is sort of independently classified as a seizure or a non-seizure activity. Now, the bulk of prior work over the last few decades has actually focused on the feature selection component. So the thought process has been, let's figure out how to extract more sensitive and specific information from this EEG, and then we can do a better job with the classification. So many areas have been explored, from like sort of Fourier domain, so time frequency analyses, to nonlinear features from nonlinear signal processing. And largely, the classification portion has been ignored. So once these features are extracted, each window is classified. Now, our strategy is to recognize that the seizure is not an isolated event, but really an evolving process. So instead of doing these independent classifications, we're actually going to try and model the spatiotemporal propagation of the seizure, or at least extract it and estimate it, and use that model to try and backtrack the onset. So our framework will rely on Bayesian models, and this is work done by my former graduate student, Jeff Crayley, at Johns Hopkins, and our collaborator, Emily Johnson, who is in the JHMI Epilepsy Center. So we can think of Bayesian models as having two parts. So first, there's the latent template, which captures unobservable phenomenon in our problem. And this is where we can inject a priori clinical assumptions or hypotheses about how the pattern should manifest. So we're actually going to use that latent template to model this spatiotemporal evolution, because of course we don't know sort of directly, or we can't say directly where it is. So we're going to try and implicitly estimate that. Now, this latent template is captured or is complemented by a set of observed data. In our cases, this is going to be either the raw EEG or features of the EEG. So that's what we can measure directly. And then all of the interactions are specified probabilistically, which allows us to infer the underlying patterns. So diving into the details, let's start with the latent template. So our problem is based on the 1020 system for EEG electrode placement. So this is the most commonly used configuration used in sort of most EEG centers worldwide, and consists of 19 channels arranged on the scalp like this. And on top, or sort of between these channels, I've drawn these blue lines. And so that's going to specify a seizure propagation graph that we're just assuming for the purposes of the model. And if you look closely, I've actually connected neighboring channels and contralateral channels, because those are the two most commonly observed spreading patterns just clinically. So we assume that all channels start in baseline, so they're all green here. So the patient is not having a seizure to start. And then as time proceeds, at some point a channel will turn on, meaning it'll start to manifest seizure activity. And this is captured by that little red channel in the bottom left. And then over time, we expect the seizure activity to spread across the graph and slowly recruit more and more channels to that seizure state. Now, of course, we don't know where or when this pattern will start, but we actually want to extract sort of patterns and information that looks like this, that kind of captures that spreading pattern through the brain.
The channels are not G channels.
They are, yeah. Because we only have scalp, so it's at the resolution of EEG channels.
I was thinking maybe this was like a graph representation, some other space, just because EEG sees a large part of the brain.
Yes, yes,
right? So,
yeah. I think with scalp-based models, that'll always be a problem. I mean, people try to do like inverse source localization, but with 19 channels, your inverse source is basically going to be a lobe. But even that is tough in this application domain. Okay, so formally, we capture that spreading using a coupled hidden Markov model. So where the state of each channel will be at the next timestamp is influenced by the current states of its neighbors. So if we focus on this little gray channel, T5, in the bottom left, in this case, all of its neighbors are in a baseline state, so they're all green. So we expect a low probability of it transitioning to a seizure state in the next time step. However, if some of its neighbors are in a seizure state, it might have a slightly higher probability of transitioning. And then if all of its neighbors are in a seizure state, then we really expect in the next time step, it will also start to manifest seizure activity. So going back to the model overview, so now that we've kind of specified the underlying assumptions and loose mathematics of that latent template, we can turn to the likelihood for the observed data. Now, as we saw on the first, one of the first slides, EEG is quite complex. So there are a lot of baseline patterns that look like they might be seizure and seizure activity that really looks like baseline. So rather than try and extract specific features and impose a certain model on them, like a Gaussian likelihood, we're actually going to use convolutional neural networks to implicitly learn a likelihood, right? And so there is a caveat to this formulation. So that's because by construction, a CNN is a posterior inference engine. So if you think about how a neural network is trained, you feed in a little bit of data, so like a snippet of EEG-Y, and the network spits out a classification. So it effectively spits out a posterior, the probability of this unknown state given your data. Now, for the Bayesian model, we want the likelihood, which is the probability of the data given the unknown state. And we can use Bayes' rule to obtain this factor. So specifically, the likelihood, Py given x, is equal to, on the numerator, the CNN output times the probability of the data itself divided by the seizure prior. And in fact, we're eventually going to run some sort of inference on that. And this Py term is constant for both states that we care about, so we can ignore it. And we basically just need that proportional term. So we need the output of the CNN, and we can sort of empirically approximate the denominator just based on the baseline seizure distribution temporally of the training data set. So combining everything together, we derive a variational EM algorithm to identify that spatiotemporal evolution as well as any of the other model parameters. So we evaluate this deep generative hybrid approach on two clinical EEG data sets. The first was an in-house data set consisting of 15 patients evaluated at Johns Hopkins. They're evaluated over several days, so there's 90 seizures total across these patients. And then the second data set is a publicly available data set think the data was collected at Boston Children's Hospital, and then it was released through MIT. It consists of 24 patients and 185 seizure recordings. So we're going to compare this deep generative hybrid approach with two baselines where we're basically decoupling the two different portions of the model. So the first baseline is just taking that sort of spreading pattern and then putting, sort of extracting specific features from the EEG and imposing a Gaussian likelihood. And then the second is throwing away that spreading model and just seeing, can we build a neural network or a CNN that classifies seizure versus baseline? So here's the testing performance from a 5-fold cross-validation. And just to parse that wall of numbers, I've just highlighted a few key trends. So we can see that our deep generative hybrid almost uniformly outperforms. And that suggests that we're really bringing together the best of both worlds. So that latent template gives us a structured model to make sense of the noisy EEG, and the CNN automatically figures out like what are the most informative features that I should be learning from this data. The other cool thing about our spreading model is that we can actually start to use it to tease out localization information. So here I've displayed 2 seizures from 2 different patients in the testing fold, and this red sort of map that you see appearing is over time the sort of the posterior probability of seizure activity in that particular location. So first thing to note is our model is able to capture different spreading patient patterns for different patients, which is always a great sanity check. And also in these cases, if we compare the onset, so the earliest occurrence of seizure activity, it does correspond to what our clinicians annotated manually as being the onset of the seizure. So moving from this model, we've started to focus more on that localization aspect, which is actually more valuable than detection for scalp EEG. So our first iteration was to extend our Bayesian model by adding this top chain or meta chain. So that top chain looks at all of the EEG data across all the channels simultaneously and just makes a determination of, is the patient starting to have a seizure? And once that onset is detected, it initiates the spreading model. So that work appeared in McKay a few years ago, and it was the first demonstration of simultaneous detection and localization from scalp EEG. We're also exploring deep learning architectures that automatically learn the spatiotemporal activity. So this is an end-to-end seizure activity tracking model. It uses a combination of convolutional encoders, so just trying to mine information from the data, and then that spreading pattern, instead of the Bayesian approach, we're using a sort of a sequence of recurrent networks to model that evolution. And our most recent work builds off the activity tracking to predict electrode level seizure onsets from global training data. So unfortunately, when we get this data from clinicians, they label the onset as maybe sort of right temporal or right parietal, but we're trying to see, can we actually narrow it down further than that? So this uses a transformer that fuses local and global information, and then kind of a sequence of recurrent modules as well. So we validated this model on data from 2 different hospitals, and it was actually the first demonstration of cross-site generalization. So even though I focused this talk on imaging genetics and epilepsy, another key area in my lab has been connectomics, or looking at sort of networks that we can extract from fMRI and diffusion MRI. And so this work uses a resting state fMRI protocol, and the key questions revolve around sort of anomaly detection using non-negative matrix factorization to preoperative mapping using deep learning for brain tumor patients. We're also working on methods to combine both structural and functional information and trying to translate them further. And finally, I do have kind of a signal processing area of the lab. So one of those projects involves how do we automatically manipulate emotional cues in speech, and sort of use that as the foundation for developing sort of standardized paradigms to understand emotion processing deficits in certain populations. Finally, just as a quick advertisement, if you do work with EEG or a similar type of signal, so one of the undergraduate students in my lab, Danielle, developed a really cool open source visualizer. So it allows you to view the EEG data, run deep learning models on the EEG, and then visualize the outputs either on the signal or on a topological scalp plot. So before concluding, I think no work can be done in a vacuum. So there are many, many people to thank. So first and foremost, I think my students are fantastic, and they've really been the ones driving the work. So everyone underlined on this slide contributed to sort of the technical work and technical content in this presentation. And then hand in hand with the students is just a fantastic set of collaborators across many departments and institutions. And then finally, our funding sources. So to wrap up, I've described a spectrum of open problems for AI and clinical neuroscience. And in the process, I've highlighted 2 projects that tackle many of the domain challenges in different ways. And I'd say that this work is really only the beginning, because I think there's really a universe of problems that'll depend on bridging the worlds of engineering and neuroscience and medicine and biology. And so these endeavors are really what I'm excited about and hope to contribute moving forward.
That was fabulous. There's a little bit of maneuvering. So what we do, Archana, is we'll start taking questions from the in-room audience, and then we'll give people time to either post a question, which you can read, ask a question, or it might be harder for you to read it, so she might read the question.
Okay.
So questions from the room first. I start?
Sure.
So I this is a lot, and it's completely unfamiliar territory to me, so it sounds dumb, say so, but I want to make sure that I understand the genetic and imaging work. So the interpretation of what features actually end up being important, it's done much later. It's after all the feature modeling has been done, right? So you showed the slide at the end where you showed how you could try to interpret. So you get these features, and then you try to interpret them in terms of either the neurosynth or the genetic.
So I'd say the important features are learned during the sort of training process. We don't know what they are. It is just, what is the data telling us are important features. So what should actually contribute to whatever we care about? So the prediction or reconstruction. But then the interpretation is post hoc. So then we look at the features and we see, so we can do sort of qualitative things like trying to map them onto the literature. You can do quantitative things like bootstrapping to see how robust are these features across different subsets of your data. Do they generalize? Do they sort of persist? But I think that sort of is a problem a little bit in this space. I will say, if you have very strong hypotheses, the model actually is good in the sense that you can inject that. So here, we're just treating our dropout mask as something we learn automatically from the data, but you can start to impose certain penalties. So if you believe that, let's say, a particular sort of set of regions is really important, you can impose penalties for selecting other regions and try to concentrate the information on that particular subset of regions, and vice versa. So if you believe that like there's no way that anything close to the cerebellum is useful for what you're doing, you can use this model to sort of mask out the effects of the cerebellum.
So I can see how that makes sense for the imaging piece, absolutely, but I think the genetic information, how do you know what's relevant? Because you're trying to not just interpret the imaging, you're trying to interpret the imaging in the context of the genetic information. And how do you know that those genetic variations that you highlighted at the end in the yellow and all the dots, how do you know I guess I'm asking, how do you know that interaction is meaningful, interaction between imaging and genetic? It's a post hoc interpretation?
Yes, and I think it necessarily has to be, because this is all retrospective data. I mean, eventually you might run a prospective experiment in a petri dish or in sort of a preclinical model where you're probing this, but for the sort of context of what we have available in human patients and human subjects, it really is going to be post hoc.
And they are meaningful in the schizophrenia population.
I mean, if you squint and look at them, they look reasonable. And what's actually interesting is that there are some new things that crop out, which probably haven't been explored. And so you can actually do either meta-analysis of the literature, you can do these quantitative validation strategies of trying to bootstrap or trying to learn on different subsets of the data to see how robust they are in terms of what persists and not.
Go ahead.
Mhm.
How many hours of interview that is out of the state health?
Mhm. So I think sort of more important is the number of examples of seizures that you have. I think that's probably more important than just the overall hours of recordings. Because basically with these deep learning models, the more patterns you show it, the better it's able to actually understand different variability, et cetera. So I don't have an answer as to how much is enough because we always never have enough. We always have like 20 to 50 patients, but we are able to learn on that. I will say for epilepsy in particular, I'm not advocating that we use this model and then surgically resect an area of the brain. I think where some of these non-invasive modalities are helpful is because they're effectively the gatekeepers to everything else done to the patient. So if you believe the seizure is in a certain region, you're more likely to implant electrodes to get fine-grained measurements there, and then from those measurements, try and resect. But if you're wrong and you implant incorrectly, you'll never actually be correct. So the patients, no matter what you do downstream, the patients won't be cured. And so I think the most that we can do, like the most information we can get out of those non-invasive modalities, even if it's just telling the clinician, hey, we found something else, why don't you sanity check this and go back to the data or do another test or run another scan. If the end result is you're doing a resection, I think any amount that you do in the beginning is going to be beneficial to helping your outcome later on.
I was just gonna ask you,
Okay.
sorry if I missed it, but could you elaborate more on the brain body?
So that is a very, very early stage. Basically, that was, we were looking at the enteric nervous system. This was actually in a preclinical model, but the idea is that there should be sort of relationships between kind of how the gut is functioning, how the enteric nervous system looks in sort of healthy and atypical populations, and that, at least in humans, is thought to sort of guide mental disorders, et cetera. The project didn't really make it to that final stage, but what we were trying to do is from these microscopy images, how do we quantify what is the structure of the enteric nervous system? So these ganglia, they're just collection of neurons. They're all different sizes, different distributions, and different tissue types. Is there a way of like using effectively sort of computer vision type of tools to parse them, to figure out what is connected to what else, what does the network look like, et cetera?
So we have two questions online. It might be easier if they unmute themselves and ask the question. Vinod, if you're there, do you want to ask the question?
Firstly, thank you for a fantastic talk. My question has got to do with the genome to phenome matrix that your study deals with from a certain angle. And as you well acknowledged, it's an enormously complex matrix. One of the many variables being discussed that has huge complications for how you then look at the phenome manifestations is the variable of neuromodulation, among many others. So I'm just curious how, in the future, as these models develop, how do you see such complex variables fitting into the model or changing or making the models more sophisticated?
I mean, I think any amount of information that you're able to bring to bear will help in terms of how do we take this very high dimensional and noisy data set and actually gain information from it. In terms of neuromodulation, I don't know if that can be really done on these types of patients. Like we have macroscopic neuroimaging data and phenotypic data. You might be able to run something like a TMS and look at a particular region and a particular activation. It might be more relevant in the context of, again, a preclinical model where you're able to modulate different areas and sort of probe different things. And in that case, I think that more of the challenge will be the interspecies relationship rather than the integration.
Okay, thank you. That's-
There's one more question. Megan, would you be okay asking a question?
Yes. I was wondering, you mentioned implantable electrodes.
Mhm.
Has there been any previous work done that uses neural networks to interpret signals from like BCIs or implantable electrodes, and whether those genetic kind of rules still apply for your algorithms?
So to answer, I think the first part of the question, yes. So I think in the BCI community and in the sort of the community that studies these implantable electrodes and ECOG and intracranial monitoring, there's been more and more work done in using deep learning and neural networks. I'm not so much in that space because I've focused primarily on neuroimaging and sort of basic electrophysiology. The second one, second part of the question, was it, I'm sorry, I think the second part of the question I kind of spaced on.
It's okay. More like, could you apply the same algorithm to subcortical structures, and as far as translating like genetics and imaging?
I think in terms of translating genetics and imaging, you would need a different way of analyzing that really high frequency sub information you get from a subcortical recording. With that said, if you have a good way of analyzing it, so it'll be a different type of architecture. It wouldn't necessarily be this feature vector. I think we could try a similar fusion strategy.
Thank you.